Pheromones fill many additional functions. Nasonov pheromones, worker bees. Royal pheromones, bees. Calming, appeasement pheromones, mammals. Necromones, given off by a deceased and decomposing organism, consisting of oleic and linoleic acids, they allow crustaceans and hexapods to identify the presence of dead conspecifics. 36. Suckling. TAA is present in rabbit milk and seems to play a role of pheromone-inducing suckling in the newborn rabbit. 37. Categorization by type. Releaser. Releaser pheromones are pheromones that cause an alteration in the behavior of the recipient. For example, some organisms use powerful attractant molecules to attract mates from a distance of 2 miles or more. In general, this type of pheromone elicits a rapid response, but is quickly degraded. In contrast, a primer pheromone has a slower onset and a longer duration. For example, rabbit mothers release mammary pheromones that trigger immediate nursing behavior by their babies. 20. Primer. Primer pheromones trigger a change of developmental events, in which they differ from all the other pheromones, which trigger a change in behavior. They were first described in Schistocerca gregaria by Maud Norris in 1954. 38. Signal. Signal pheromones cause short-term changes, such as the neurotransmitter release that activates a response. For instance, GnRH molecule functions as a neurotransmitter in rats to elicit lordosis behavior. 5. Pheromone receptors. In the olfactory epithelium. The human trace amine-associated receptors are a group of 6G protein-coupled receptors, i.e., TAR1, TAR2, TAR5, TAR6, TAR8, and TAR9 that, with exception for TAR1, are expressed in the human olfactory epithelium. 39. In humans and other animals, TARs in the olfactory epithelium function as olfactory receptors that detect volatile amine odorants, including certain pheromones. 39. 40. These TAARs putatively function as a class of pheromone receptors involved in the olfactive detection of social cues. 39. 40. A review of studies involving non-human animals indicated that TARs in the olfactory epithelium can mediate attractive or aversive behavioral responses to a receptor agonist. 40. This review also noted that the behavioral response evoked by a TAR can vary across species, e.g., TAR5 mediates attraction to trimethylamine in mice and aversion to trimethylamine in rats. 40. In humans, HTAR5 presumably mediates aversion to trimethylamine, which is known to act as an HTAR5 agonist and to possess a foul, fishy odor that is aversive to humans. 40. 41. However, HTAR5 is not the only olfactory receptor that is responsible for trimethylamine olfaction in humans. 40. 41. As of December 2015, HTAR5-mediated trimethylamine aversion has not been examined in published research. 41. In the vomeronasal organ. In reptiles, amphibia and non-primate mammals, pheromones are detected by regular olfactory membranes, and also by the vomeronasal organ, VNO, or Jacobson's organ, which lies at the base of the nasal septum between the nose and mouth and is the first stage of the accessory olfactory system. 42. While the VNO is present in most amphibia, reptiles, and non-primate mammals, 43. It is absent in birds, adult catarrhine monkeys downward-facing nostrils, as opposed to sideways, and apes. 44. An active role for the human VNO in the detection of pheromones is disputed. While it is clearly present in the fetus, it appears to be atrophied, shrunk, or completely absent in adults. 3 distinct families of vomeronasal receptors, putatively pheromone sensing, have been identified in the vomeronasal organ named V1 rupee, V2 rupees, and V3 rupees. All are G protein coupled receptors but are only distantly related to the receptors of the main olfactory system, highlighting their different role. 42. Evolution. Olfactory processing of chemical signals like pheromones exists in all animal phyla and is thus the oldest of the senses. It has been suggested that it serves survival by generating appropriate behavioral responses to the signals of threat, sex, and dominance status among members of the same species. 45. Furthermore, it has been suggested that in the evolution of unicellular prokaryotes to multicellular eukaryotes, primordial pheromone signaling between individuals may have evolved to paracrine and endocrine signaling within individual organisms. 46. Some authors assume that approach avoidance reactions in animals, elicited by chemical cues, form the phylogenetic basis for the experience of emotions in humans. 47. Evolution of sex pheromones. Avoidance of inbreeding. Mice can distinguish close relatives from more distantly related individuals on the basis of scent signals. 48. Which enables them to avoid mating with close relatives and minimizes deleterious inbreeding. 49. In addition to mice, 2 species of bumblebee, in particular Bombus bifarius and Bombus frigidus, have been observed to use pheromones as a means of kin recognition to avoid inbreeding. 50. For example, B. bifarius males display patrolling behavior in which they mark specific paths outside their nests with pheromones and subsequently patrol these paths. 50. Unrelated reproductive females are attracted to the pheromones deposited by males on these paths, and males that encounter these females while patrolling can mate with them. 50. Other bees of the Bombus species are found to emit pheromones as precopulatory signals, such as Bombus lapidarius. 51. Applications. Pheromone trapping. Pheromones of certain pest insect species, such as the Japanese beetle, acrobat ant, and the spongy moth, can be used to trap the respective insect for monitoring purposes, to control the population by creating confusion, to disrupt mating, and to prevent further egg laying. Animal husbandry. Pheromones are used in the detection of estrus in sows. Boar pheromones are sprayed into the sty, and those sows that exhibit sexual arousal are known to be currently available for breeding. Human sex pheromone controversies. While humans are highly dependent upon visual cues, when in close proximity, smells also play a role in sociosexual behaviors. An inherent difficulty in studying human pheromones is the need for cleanliness and odorlessness in human participants. 52. Though various researchers have investigated the possibility of their existence, no pheromonal substance has ever been 52. Though various researchers have investigated the possibility of their existence, no pheromonal substance has ever been demonstrated to directly influence human behavior in a peer-reviewed study. 53, 54, 55, 56. Experiments have focused on three classes of possible human pheromones: axillary steroids, vaginal aliphatic acids, and stimulators of the vomeronasal organ, including this 2018 study. https://academic.upe.com/scan/article/14/7/719/5532402 claiming pheromones affect men's sexual cognition. Axillary steroids. Axillary steroids are produced by the testes, ovaries, apocrine glands, and adrenal glands. 57. These chemicals are not biologically active until puberty, when sex steroids influence their activity. 58. The change in activity during puberty suggests that humans may communicate through odors. 58. 57. These chemicals are not biologically active until puberty, when axillary steroids are produced by the testes, ovaries, apocrine glands, and adrenal glands. 57. These chemicals are not biologically active until puberty, when sex steroids influence their activity. 58. The change in activity during puberty suggests that humans may communicate through odors. 57. Several axillary steroids have been described as possible human pheromones: androstadienol, androstadienone, androstenol, androstenone, and androsterone. Androstenol is the putative female pheromone. 58. In a 1978 study by Kirk Smith, people wearing surgical masks treated with androstenol or untreated were shown pictures of people, animals, and buildings and asked to rate the pictures on attractiveness. 59. Individuals with their masks treated with androstenol rated their photographs as being warmer and more friendly. 59. The best-known case study involves the synchronization of menstrual cycles among women based on unconscious odor cues, the McClintock effect, named after the primary investigator, Martha McClintock, of the University of Chicago. 60. 61. A group of women were exposed to a whiff of perspiration from other women. Depending on the time in the month the sweat was collected before, during, or after ovulation, there was an association with the recipient woman's menstrual cycle to speed up or slow down. The 1971 study proposed two types of pheromone involved. 1. Produced prior to ovulation, shortens the ovarian cycle, and the second, produced just at ovulation, lengthens the cycle. However, recent studies and reviews of the methodology have called the validity of her results and existence of menstrual synchronization into question. 62. 63. Androstenone is postulated to be secreted only by males as an attractant for women, and thought to be a positive effector for their mood. It seems to have different effects. 62. 63. Androstenone is postulated to be secreted only by males as an attractant for women, and thought to be a positive effector for their mood. It seems to have different effects on women, depending on where a female is in her menstrual cycle, with the highest sensitivity to it during ovulation. 58. In 1983, study participants exposed to androstenone were shown to undergo changes in skin conductance. 64. Androstenone has been found to be perceived as more pleasant to women during their time of ovulation. 52. Androstadienone seems to affect the limbic system and causes a positive reaction in women, improving mood. 57. Responses to androstadienone depend on the individual and the environment they are in. 65. Androstadienone negatively influences the perception of pain in women. 65. Women tend to react positively after androstadienone presentation, while men react more negatively. In an experiment by Hummer and McClintock, androstadienone or a control odor was put on the upper lips of 50 males and females, and they were tested for 4 effects of the pheromone. 1. Automatic attention towards positive and negative facial expressions. 2. The strength of cognitive and emotional information as distractors in a simple reaction time task. 3. Relative attention to social and non-social stimuli, i.e. neutral faces. And 4. Mood and attentiveness in the absence of social interaction. Those treated with androstadienone drew more attention towards emotional facial expressions and emotional words, but no increased attention to neutral faces. These data suggest that androstadienone may increase attention to emotional information, causing the individual to feel more focused. It is thought that androstadienone modulates on how the mind attends and processes information. 65. While it may be expected on evolutionary grounds that humans have pheromones, these 3 molecules have yet to be rigorously proven to act as such. Research in this field has suffered from small sample sizes, publication bias, false positives, and poor methodology. 66. Vaginal aliphatic acids. A class of aliphatic acids, volatile fatty acids, is a kind of carboxylic acid, was found in female rhesus monkeys that produced 6 types in the vaginal fluids. 67. The combination of these acids is referred to as copulins. One of the acids, acetic acid, was found in all of the sampled females' vaginal fluid. 67. Even in humans, 1/3 of women have all 6 types of copulins, which increase in quantity before ovulation. 67. Copulins are used to signal ovulation. However, as human ovulation is concealed, it is thought that they may be used for reasons other than sexual communication. 57. Stimulators of the vomeronasal organ. The human vomeronasal organ has epithelia that may be able to serve as a chemical sensory organ. However, the genes that encode the VNO receptors are non-functional pseudogenes in humans. 52. Also, while there are sensory neurons in the human VNO, there seem to be no connections between the VNO and the central nervous system. The associated olfactory bulb is present in the fetus, but regresses and vanishes in the adult brain. There have been some reports that the human VNO does function, but only responds to hormones in a sex-specific manner. There also have been pheromone receptor genes found in olfactory mucosa. 52. There have been no experiments that compare people lacking the VNO and people that have it. It is disputed on whether the chemicals are reaching the brain through the VNO or other tissues. 57. In 2006, it was shown that a second mouse receptor subclass is found in the olfactory epithelium. Called the trace amine-associated receptors, TAR, some are activated by volatile amines found in mouse urine, including one putative mouse pheromone. 68. Orthologous receptors exist in humans, providing, the authors propose, evidence for a mechanism of human pheromone detection. 69. Although there are disputes about the mechanisms by which pheromones function, there is evidence that pheromones do affect humans. 70. Despite this evidence, it has not been conclusively shown that humans have functional pheromones. Those experiments suggesting that certain pheromones have a positive effect on humans are countered by others indicating they have no effect whatsoever. 57. A possible theory being studied now is that these axillary odors are being used to provide information about the immune system. Malinsky and colleagues found that the artificial odors that people chose are determined in part by their major histocompatibility complexes, MHC combination. 71. Information about an individual's immune system could be used as a way of sexual selection, so that the female could obtain good genes for her offspring. 52. Klaus Wedekind and colleagues found that both men and women prefer the axillary odors of people whose MHC is different from their own. 72. Some body spray advertisers claim that their products contain human sexual pheromones that act as an aphrodisiac. Despite these claims, no pheromonal substance has ever been demonstrated to directly influence human behavior in a peer-reviewed study. 57. 55. Thus, the role of pheromones in human behavior remains speculative and controversial. 73. See also: aphrodisiac, allomone, ant mill, cat. Insect pheromones. Insect pheromones are neurotransmitters that serve the chemical communication between individuals of an insect species. They thus differ from chiromones, in other words, neurotransmitters that transmit information to non-species organisms. Insects produce pheromones in special glands and release them into the environment. In the pheromone receptors of the sensory cells of the recipient, they produce a nerve stimulus even in very low concentrations, which ultimately leads to a behavioral response. Intraspecific communication of insects via these substances takes place in a variety of ways and serves, among other things, to find sexual partner, to maintain harmony in a colony of socially living insects, to mark territories or to find nest sites and food sources. 500,000 scent glands of the female silkmoth, Bombyx mori, were needed to elucidate the molecular structure of bombicol. 1. Structural formula of bombicol, the first clearly chemically identified insect pheromone. In 1959, the German biochemist and Nobel Prize winner Adolf Butenandt identified and synthesized the unsaturated fatty alcohol bombicol, the sex pheromone of the domestic silk moth Bombyx mori, as the first known insect pheromone. The sex pheromones of female butterflies are mostly mon- or bisulfinic fatty acids or their esters, fatty alcohols, their esters are the corresponding aldehydes. Male butterflies use a wide range of chemicals as sex pheromones, for example pyrrolizidine alkaloids, terpenes and aromatic compounds such as benzaldehyde. Research into the chemical communication of insects is expanding our understanding of how they locate their food sources or places to lay eggs. For example, beekeepers use an artificially produced Nasanov pheromone containing terpenes such as geraniol and citral to attract bees to an unused hive. The agriculture and forestry industries use insect pheromones commercially in pest control using insect traps to prevent egg laying and in practicing the mating disruption. In 1959, the German biochemist and Nobel Prize winner Adolf Butenandt identified and synthesized the unsaturated fatty alcohol bombicol, the sex pheromone of the domestic silk moth Bombyx mori, as the first known insect pheromone. The sex pheromones of female butterflies are mostly mon- or bisulfinic fatty acids or their esters, fatty alcohols, their esters are the corresponding aldehydes. Male butterflies use a wide range of chemicals as sex pheromones, for example pyrrolizidine alkaloids, terpenes and aromatic compounds such as benzaldehyde. Research into the chemical communication of insects is expanding our understanding of how they locate their food sources or places to lay eggs. For example, beekeepers use an artificially produced Nasanov pheromone containing terpenes such as geraniol and citral to attract bees to an unused hive. The agriculture and forestry industries use insect pheromones commercially in pest control using insect traps to prevent egg laying and in practicing the mating disruption. It is expected that insect pheromones can also contribute in this way to the control of insect-borne infectious diseases such as malaria, dengue fever or African trypanosomiasis. Etymology and classification. Adolf Butenandt and Peter Carlson proposed the term pheromones in 1959 for substances that serve intraspecific communication. 2. The definition of the term pheromone was given in the same year by Carlson and the Swiss zoologist Martin Luscher. According to this, pheromones are substances released externally by one individual that elicit specific responses in another individual of the same species. Peter Carlson, Martin Luscher, 1959. 3. The word pheromone consists of the ancient Greek parts of speech pherein pherein, überbringen, melden, and hormon hormon, antrieben, erregen. 3. 4. According to Carlson and Luscher, the goal was to coin an internationally understandable scientific term for a class of substances based on a clear definition. It was to be a short word that could be spoken in many languages. The ending -mone served as a suffix, as it occurs in the words hormone, chiromone, and allomone, thus emphasizing their relationship. 5. The term pheromone replaced the term ectohormone or homoiohormone, which Albrecht Beta had already proposed in 1932 with the same definition. 6. Beta's term was not accepted because, according to Boudinant, the terms ecto and hormone were mutually exclusive. The mechanism of action of a pheromone also does not correspond to that of a hormone absorbed into the circulatory system by another individual and was therefore considered misleading. The classification of intraspecific pheromones in the group of semiochemicals, in other words, neurotransmitters that serve communication between organisms, is shown in the following diagram. 7. Carlson further divided them into the sense of smell and the oral-acting insect pheromones according to the mode of reception. 8. In 1963, Edward O. Wilson, who had discovered ant-trace pheromones the year before, and William H. Bossert introduced the concepts of releaser and primer pheromones. 9. 10. Releaser pheromones, which are usually perceived olfactorily, cause an instantaneously observable behavioral reaction, whereas primer pheromones, which are often oral, trigger physiological changes in the recipient. Primer pheromones, for example, suppress the formation of ovaries in worker bees. Often, pheromones are defined according to their behavior triggering function. In addition to the well-known sex attractants, they act, among other things, as aggregation pheromones, dispersion pheromones, alarm pheromones, tracking pheromones, marker pheromones, brood recognition pheromones, egg-laying pheromones, recruitment pheromones, or as caste recognition agents. Vincent de Thier divided insect pheromones into 6 categories according to their general behavior triggering effects. 11. These include prisoners, which are normally only perceptible at short distances and cause an insect in motion to stop, and locomotor stimulants, which increase the insect's speed or decrease the number of directional changes. Lockstop are attractants that trigger an oriented movement toward the odor source, whereas repellents trigger an escape movement away from it. Feeding and oviposition stimulants, respectively, trigger feeding or oviposition. Deterrents, on the other hand, inhibit feeding or oviposition. Functionally defined insect pheromones often contain mixtures of different components in precisely defined proportions. These so-called pheromone cocktails often contain substances of different categories with near and far orientation functions. For example, the aggregation pheromone cocktail of the German cockroach Blattella germanica contains both substances that act as attractants and substances that act as arrestants. 12. In part, insect pheromones are named after the site of their biological production. Males of various moth species, such as the banana butterfly, possess so-called androconial organs in the abdomen that release pheromones. These insect pheromones are appropriately called androconial pheromones. 13. The queen bee of the western honeybee produce the queen bee pheromone in mandibular glands. In English, they are therefore often referred to as queen mandibular gland pheromones. 14. History. First discoveries. John Ray. In 1609, the English beekeeper Charles Butler observed that the sting of a bee released a liquid. This liquid attracted other bees, which then began to sting en masse. 15. Butler thus demonstrated for the first time the effect of an alarm pheromone of bees, which was identified as isoamyl acetate in the 1960s. 16. Butler's observation was the first to show the effect of an alarm pheromone of bees, which was identified as isoamyl acetate in the 1960s. As early as 1690, Sir John Ray suspected that peppered moths attracted male conspecifics by means of a scent. It emerged out of a stick-shaped geometer caterpillar. It was a female and came out from its chrysalis shut up in my cage. The windows were open in the room or closet where it was kept, and 2 male moths flying round were caught by my wife who by lucky chance were into the room in the night. They were attracted, as it seems to me, by the scent of the female and came in from outside. It developed from a stick-shaped birch moth caterpillar. It was a female, and came out of her chrysalis, which was shut up in my cage. The windows were open in the room or chamber where she was kept, and 2 male moths flying about were caught by my wife, who by lucky chance was in the room that night. They were attracted, it seems to me, by the scent of the female, and came in from outside. John Ray, 17. Jean-Henri Fabre. The French entomologist Jean-Henri Fabre also reported in the mid-19th century on experiments with Saturnia and oak egger in which females trapped in wire cages attracted hundreds of males within a few days at specific times. 18. In experiments with tagged domestic silk moth, 40% of males from a distance of 4 kilometers and 26% of males from 11 kilometers still found their way to a trapped female. 18. The morphology of the moths was also reported in the mid-19th century. In many insect species, researchers long puzzled over the mechanism of mating. Visual or acoustic stimuli could not explain Fabre's experiments, nor how moths found females ready to mate with great certainty. Theories of attraction by infrared or other radiation were not confirmed. 18. The organization of eusociality remained equally inexplicable for a long time. The writer and bee researcher Maurice Maeterlinck speculated about the spirit of the hive, the teemed spirit of the hive, without being able to determine its essence in more detail. 19. Theories about the attraction by infrared or radiation also remained unexplained for a long time. Definitions of beta. At the beginning of the 20th century, Ernest Starling discovered hormones as the first biological neurotransmitters. 20. In 1932, the neurophysiologist Albrecht Beta, who at that time headed the Institute of Animal Physiology at the Goethe University Frankfurt, published an article on an expanded hormone concept in which he distinguished between endohormones and ectohormones. 6. According to this, endohormones act in the producing organism itself and correspond to the classical hormone definition. In contrast, the organism releases ectohormones externally and transfers them to other individuals. As an example, Beta cited the effect of the lactation hormone, which is released by a fetus to its mother and causes the growth of the mammary gland and subsequently the secretion of milk. 6. He also proposed this concept for chemical communication among insects. In bees, for example, the workers, i.e. not the mothers, are able to raise a sexually capable queen from an egg or a young larva by special food and transfer of secretions of their salivary glands. There can hardly be any doubt, although it has not been proven, that ectohormones of the salivary gland secretions play the main role in this redifferentiation. Albrecht Beta, 6. Beta further divided the ectohormones into homoiohormones, which, according to today's definition of a pheromone, act on individuals of the same species, and alloiohormones, which act on individuals of a different species. He thus coined the precursor term of allochemicals. 6. Works from Boudinant. Adolphe Boudinant also suspected that communication among insects was based on neurotransmitters, and in the 1940s he began a project to identify the sexual attractant of the domestic silk moth, Bombyx mori. It is a butterfly originally native to China, belonging to the family of the Bombycidae, which is used for sericulture, and its breeding and keeping was well known. It was only after almost 20 years of work that he finally succeeded in extracting and purifying a substance from more than 500,000 insects, which Boudinant later named Bombycol. By elemental analysis, Boudinant determined the chemical formula of the substance to be C16H30O. Infrared spectroscopic studies indicated the presence of conjugated double bonds. Using methods common at the time, such as catalytic hydrogenation, melting point determination, and oxidative degradation by potassium permanganate, Boudinant showed that the substance sought was an unsaturated fatty alcohol, 10E, 12Z-10, 12HEXADECADIEN1OL. 21. Boudinant then synthesized Bombycol from bernolic acid, 12R, 13S, epoxy 9-cis-octadecanoic acid, in several steps via diol formation, its cleavage to the aldehyde, double bond isomerization, and Wittig reaction. He synthesized the 4 possible stereoisomers and tested them for their biological activity. 22. Only one isomer showed the same activity as the extract. Boudinant thus provided evidence that communication among insects takes place on a substance-by-substance basis. By extraction and condensation experiments, however, it has been convincingly shown that a material principle must be present which is secreted by the female butterflies from scent organs of the last abdominal segments and perceived by the males with their antennae. Adolphe Boudinant, 23. Primer and releaser pheromones. E. O. Wilson. Towards the end of the 1950s, Edward O. Wilson defined substances that trigger the alarm and burrowing behavior of ants as chemical releaser. 24. In 1961, the British biochemist Robert Kenneth Callow identified another pheromone, also known as the queen bee pheromone, with the compound E-9-oxodec-2-enoic acid, or 9-ODA for short. 25. The effect of this pheromone was obviously of a different nature than that of the alarm pheromones, since it had a long-term effect on the physiology of the recipients. In 1963, Wilson, who the year before had already discovered the trace pheromones of ants, and William H. Bossert introduced the term releaser and primer pheromones for this purpose, to distinguish the behavior-controlling effect of, for example, sex attractants from the pheromones that interfere with the endocrine system of the receiver. 9-10. Modern research directions. Preparation of Helicoverpa Z. As a result of the enormously refined extraction and analytical chemistry over the years, chemists and biologists identified numerous other pheromones. For the detection of the second component of the pheromone cocktail of Bombyx mori, the Bombycal 10Z, 12E hexadecadienyl, an extract of 460 glands from which 15 nanograms of the aldehyde were isolated was already sufficient in 1978. 26. In addition to studying the function and reception of pheromones and their chemical identification, scientists extensively investigated the biochemistry of pheromone production. In 1984, Ashok Raina and Jerome Klun discovered that the production of the female sex attractant of the owlet moths Helicoverpa Z is controlled by hormonal substances called pheromone biosynthesis activating neuropeptides (PBANs) in the brains of female moths. 27. Other modern research focuses on the study of insect pheromone reception by means of the sense of smell and taste, genetic factors, and evolutionary biology issues, such as the coevolution of female sex pheromone production and reception in the male. 28. Combating disease vectors such as anopheles is another focus of research. According to World Health Organization estimates, the number of malaria infections in 2012 was about 207 million with 627,000 deaths. 29. Culex mosquitoes transmit the causative agent of filariasis or West Nile virus. Traps equipped with oviposition pheromones offer one way to contain these populations. To optimize these, the scent-binding proteins in the antennae of females, which play a critical role in detecting oviposition sites, are being intensively studied. 30. Production. The pheromones in insects are often the secondary products of fatty acids, such as saturated and unsaturated hydrocarbons, fatty alcohols, esters and aldehydes, but also isoprenoids and other compounds. Pheromones are often not pure substances, but so-called pheromone cocktails consisting of various components. Often, only one specific enantiomer of a compound triggers a behavioral reaction, while the other enantiomer triggers no reaction or a different reaction. Sometimes the biosynthesis of the pheromone occurs only when the biochemical precursors in the form of certain alkaloids have been ingested from food plants. In this case, the sex pheromone simultaneously signals the presence of food sources. 31. Due to the potential commercial application in crop protection, the intensity of the study of pheromones increased greatly after Boudinant's discovery, leading to the development of highly sensitive analytical methods 32 and the widespread application of chemo, regio, and stereoselective syntheses in organic chemistry. Biosynthesis. Male of silver-washed fritillary with 4 elongated scent scale patches, scent scale stripes, on each forewing. Insect pheromones are produced by a variety of exocrine glands consisting primarily of modified epidermal cells at various sites on the insect body. For example, the abdominal glands of the female silkmoth release traces of the E isomer of alcohol as well as the analogous Z-aldehyde bombical, in addition to the sex pheromone bombical. 33-34 Suitable surface geometries in the vicinity of the glands, such as grooved pore plates, may favor effective evaporation of a leaked pheromone. 35 Honeybees possess 15 glands with which they produce and release a number of different substances, thus maintaining a complex communication system based on pheromones. 36-37 Males of various butterfly species possess so-called androconial organs in the abdomen with which they can disseminate pheromones, while other moths release them via scent shed or scent bristles on their forewings or the end of the abdomen. The scent bristles and scent scales serve to increase surface area and facilitate the evaporation of insect pheromones. 35 Rather than evolving a completely unique set of enzymes for pheromone biosynthesis, insects often modify normal metabolites into pheromones with high regio, chemo, Z diestereo, or enantioselectivity and in well-defined ratios. 38 Biosynthesis of insect pheromones occurs either de novo following the scheme of fatty acid synthesis by successive addition of malonyl-CoA to an initial acetyl or by uptake of precursors from the diet. Many butterflies use the biosynthetic possibility of producing a specific mixture of derivatives of simple fatty acids. The evolution of the enzyme delta-11 desaturase combined with chain-shortening reactions allows them to produce a variety of unsaturated acetates, aldehydes, and alcohols in various combinations. 39 Dehydrogenation of the carbon chain and reduction of the acid function to the alcohol occurs, if necessary, through special enzyme systems. Further steps can be oxidation to the aldehyde or acetylation to the acetate. 40 In Bombyx mori, the biosynthesis is activated diurnally by pheromones through a neurohormone, the so-called pheromone biosynthesis activating neuropeptide PBAN. 40 The pheromone biosynthesis activating neuropeptide PBAN is a neurohormone. The hormonal mechanisms of pheromone production vary considerably from species to species. 41 Juvenile hormones, for example, control pheromone production in the owl butterfly Mithymna unipuncta. These are produced in mostly paired corpora allata located behind the brain and released into the hemolymph. There they bind to certain transport proteins. If the corpora allata are removed, the females do not produce pheromones. Juvenile hormones, however, interfere more indirectly with the circadian release of PBAN. 42 Male butterflies of the Daninae family sometimes wound caterpillars that have ingested alkaloids from milkweeds with tiny claws on their feet to ingest the exuding fluid, a behavior described as kleptopharmacophagy. The moths use the ingested alkaloids for defense against predators and to produce sex pheromones. 43 Pheromones from plant ingredients. Retronacine, from which many pyrrolizidine alkaloids are derived by esterification of the 2-hydroxyl groups. Male fire-colored beetles of the species Neopyracroa flavulata and also various other beetle species use the terpenoid cantharidin as a sex pheromone and aphrodisiac pheromone, respectively. This isoprenoid is ingested by Neopyracroa flavulata with food and transferred to females and subsequently to the brood during the mating act. 44 Females test the content of a gland on the head of the male before mating. The cantharidin acts as a feeding toxin and renders the eggs unpalatable to predators. Females therefore prefer males with a high cantharidin content. 47 Moths such as Euthethysa ornatrix and Tyriamala limnaeus ingest pyrrolizidine alkaloids from food plants such as Crotalaria, Heliotropium, or Achillea ageratum in the caterpillar, which the adult male converts by oxidation into pheromones such as hydroxydanidyl. As in the fire beetle, the alkaloids, which are potent feeding poisons and act against predators such as spiders, ants, or net-winged insects, are transferred to females and eggs. 45 Adult monarch butterflies ingest plants' secondary metabolism to increase their pheromonal attractiveness. 46 Sometimes biosynthesis of the pheromone occurs only when biochemical precursors in the form of certain alkaloids have been ingested from food plants. In this case, the sex pheromone simultaneously signals the presence of food sources. 47 The uptake of pheromone precursors from plants is also known for certain species of orchid bees and peacock flies. Male bees collect a mixture of terpenoids from orchids and use them as an aggregation pheromone to form lek mating. Sometimes the plant constituents control the development of the pheromone glands of male butterflies. 48 Laboratory synthesis. Carl Ziegler and Gunther Otto Schenk succeeded in synthesizing cantharidin as early as 1941. Carl Ziegler and Gunther Otto Schenk succeeded in synthesizing cantharidin as early as 1941. 4950 The preparation of pheromones requires the use of highly chemo, regio and stereoselective syntheses. In the 1970s, asymmetric synthesis using the SAMP method succeeded in producing various pheromones enantiomerically pure. 51 Furthermore, chemists used asymmetric epoxidation, asymmetric dihydroxylation, biocatalysis, olefin metathesis, and many other stereoselective reactions to synthesize pheromones. 52 The Wittig reaction is suitable for the chemical synthesis of pheromones with Z-olefinic double bonds. 53 Genetically modified tobacco plants can also produce sex pheromones. The fatty alcohols obtained from them by extraction are subsequently acetylated to obtain the respective target sex pheromones. This semi-synthetic route of production produces insect pheromones in relatively large quantities and with high purity. 54. Properties. Chamaespecia empiformis on pheromone dispenser. Chemical communication between living beings by means of pheromones follows the same principles as technical data transmission. A transmitter, for example the gland of a female insect, emits the signal in the form of a chemical substance. Both the chemical structure of the molecules and their quantity ratio determine the information content and serve as a model of communication for the species. The physical properties of the substances, such as vapor pressure, determine the function of their molecules as shorter long-range information transmitters. 55. The insect pheromone is transmitted by direct contact or via a medium such as water or air. From the receiver, for example the pheromone receptors in the antenna of a male insect, the substance is received and triggers a behavioral response. The term antenna was first used to refer to the antennae of insects and subsequently in engineering. 56. Insect pheromones have a highly specific effect, meaning that they elicit the desired behavioral response only in biological specificity but not in individuals of other species. For example, although the chemical compounds that act as sex pheromones in butterflies may be the same in different species, the composition of the pheromone cocktail is different in all species. 55. In addition, pheromone cocktails often contain substances that act as behavioral inhibitors for other species, such as significantly reducing the rate of approach of males of alien species to an attracting female. 56. The pheromone cocktail is a highly effective means of inhibiting the behavior of males of other species. Physico-chemical properties. The pheromones are usually produced as a liquid and are either transmitted by direct contact or released into the environment as a liquid or vapor. They can be either heavy or light volatile. Diffusivity significantly affects the function of the pheromone. 57. Alarm pheromones are often highly volatile to spread quickly by diffusion. Therefore, they are often short-chain substances with relatively high vapor pressure and low complexity. 40. There is no high requirement for specific coding effect as in sex pheromones. Sex pheromones have a higher complexity than most alarm pheromones, but a lower molar mass than marker pheromones, which permanently indicate an area. 58. In the case of flying insects such as butterflies, the pheromone as a molecule must not be too large, otherwise the vapor pressure and volatility are too low. Thus, over 200 identified sex pheromones of butterfly species are mono- and bisulfinic fatty aldehydes, fatty alcohols and their acetates with chains of 10 to 18 carbon atoms. 40. Depending on the function, there are different emission and reception scenarios. Ants, for example, emit alarm pheromones intermittently or continuously in the usually windless environment of the anthill. Trace pheromones are emitted by an ant as a moving source. Silkmoth sex pheromones are emitted in discrete scent threads in an air stream. 57. Monarch butterfly male with clearly visible scent scales pockets. Male monarch butterflies do not emit volatile pheromones, but pheromone-laden nanoparticles called pheromone transfer particles, which they use to transfer aerostants or aphrodisiac pheromones to females. The pheromone transfer particles position the males on their brush hairs and scatter them during courtship flight. The nanoparticles adhere to the antennae of the females, which are equipped with pheromone receptors, where they slowly release the pheromones, resulting in a long-lasting stimulus for the female. 59. Females of the Arctiinae Pyrrharctia isabella emit an aerosol consisting exclusively of sex pheromone droplets. The amount of pheromone released in this process is much greater than in other known female moths. The apparent wastefulness of the sex pheromone is explained by the short amount of time an adult has to find a reproductive partner due to the short Arctic spring. 60. Recipients usually perceive pheromones in an environment characterized by the presence of many other chemicals. To ensure specific perception, the pheromone chemical must either be so complex that it does not occur more than once in nature, or the correct ratio of several individual components must trigger the stimulus. However, it has been shown that only in exceptional cases does a single substance convey the message. Often, a mixture of substances must be present in very precise proportions that, in addition to the chemical structure of the individual pheromones, determine the informational content of the pheromone cocktail. 40. Paraplanin B. The chemical structure of pheromones is directly related to their signaling function and signaling environment. Pheromones released into the air often have a carbon chain of 5 to 20 atoms and a molar mass of about 80 to 300 grams mol1. With a carbon chain of less than 5 carbon atoms, the number of possible isomers is small and targeted species-specific coding is difficult. 58. With longer carbon chains, the number of possible isomers increases rapidly. Paraplanin B, the sex pheromone of the American cockroach, is an example of a complex single substance to which males respond at extremely low levels of 10-5 nanograms. 61, 62. Biological properties. Antenna of Elasiocampidi. The sex pheromone cocktail emitted by a female insect spreads downwind. In the recipient male, the molecules strike the antennae, where reception of the pheromones takes place by means of olfactory cells on the olfactory hairs or cincilla. The antennae absorb about 30% of the pheromone molecules contained in an airstream. 56. The remaining molecules hit the outer exoskeleton where they are enzymatically degraded. The pheromone molecules first reach the cuticle of the olfactory hairs and diffuse through pores into a pore cone and from there into tubules. From there, the molecules diffuse further to the dendritic membrane. 40. This membrane has receptors that, when a pheromone is received, cause a change in electrical resistance via the opening of ion channels, creating an electrical resistance that results in a perception. 40. Even a single pheromone molecule can trigger a nerve impulse. 59. However, the recognition of a specific pheromone cocktail requires a certain level of excitation of different cell types of varying specificity. 56. It is assumed that the characteristic excitations received from the different receptors in the central nervous system are modulated there into an excitation pattern. If this excitation pattern, which depends on the quantitative ratio of the received pheromone molecules, matches the coding of an innate behavioral pattern, this leads to the triggering of a corresponding behavioral response, such as the headwind approach to a pheromone source. 55. Pheromone species. According to their effect, two classes of pheromones can be distinguished, the primer and the releaser pheromones. Under certain conditions, certain pheromones act as both releaser and primer pheromones. 63. Releaser pheromone. Releaser pheromones have a brief, immediate behavior controlling effect. The first pheromone discovered, bombicol, is an example. Releaser pheromones typically include aggregation pheromones, dispersal pheromones, alarm pheromones, tracking pheromones, and marking pheromones, among others, in addition to the well-known sex pheromones. 63. Aggregation pheromones. Aggregation of pentatonidy. Aggregation pheromones are produced by both sexes and are used for the sex-nonspecific attraction of individuals of the same species. These are known, for example, in the bark beetle and other beetle species, bipeds, hemiptera and grasshoppers. Insects use aggregation pheromones for defense against predators, in mate choice, and to overcome host plant resistance to mass attack. A group of individuals at a site is referred to as an aggregation, regardless of sex. 64. Aggregation pheromones, in addition to sex attractants, play a significant role in the development of pheromone traps for selective pest control. 65. Studies using electroantennogram techniques showed that aggregation pheromones elicited relatively high receptor potentials in a sex-nonspecific manner, whereas sex pheromones elicited high receptor potentials in only one sex. Aggregation pheromones may therefore be evolutionary precursors of sex pheromones. 35. Sex pheromones. Copulating poplar hawk moth pair. Sex pheromones signal the female animal's readiness to mating. Male animals also emit pheromones. They contain information about sex and genotype. Many insects release sex pheromones. Some butterfly species still perceive the pheromone at a distance of 10 kilometers. The sensory cell response in the male silkmoth begins at a concentration of about 1,000 molecules per cubic centimeter of air. 56. The scent signal of a female, as soon as a certain concentration threshold is exceeded, initially triggers an oriented headwind flight in the silkmoth male. 56. In other species such as the codling moth, on the other hand, the male tests the stereochemical purity of the attractant molecule. As soon as a small admixture of another stereoisomer is present in the pheromone cocktail, the approach to the source stops. 56. In this case, the other stereoisomer acts as a repellent. 40. In addition to the main components, some species release so-called short-range components in small amounts, which influence the behavioral response. 56. In addition to the main components, some species release so-called short-range components in small amounts. Foraging honeybees spread the scent of Z-11 EICOSEN10L. European bee wolves are guided by this scent to prey on honeybees. Male bee wolves use this component, and thus the existing sensory preference of females for bee scent, as part of their sex pheromone cocktail to attract them. 66. Aphrodisiac apheromones. Olein, the aphrodisiac apheromone of the olive fruit fly, Bactrocera olei. Aphrodisiac pheromones stimulate the readiness to mate. The spiroacetyl olein, for example, is the aphrodisiac pheromone of the olive fruit fly, Bactrocera olei. Only the R enantiomer is effective on males, the S enantiomer is ineffective on them. The female produces the racemate, responds to R and S olein, and also stimulates herself with it. 67. So-called anti-aphrodisiacs have exactly the opposite effect. Bedbug nymphs protect themselves with such a pheromone, which has a specific mixing ratio of the aldehydes E-2-hexenyl, E-2-octenyl and 4-oxo E-2-hexenyl, against mating attempts by male bedbugs. The latter directly drills a hole into the abdomen of sexually mature female bedbugs and injects its sperm their traumatic insemination. For mated nymphs, however, such injury can be fatal. 68. Alarm pheromones. Some insect species emit alarm pheromones when attacked. These trigger either flight or increased aggression. In bees, for example, two alarm pheromone mixtures are known. One is released by the Koshevnikov gland near the sting and contains more than 40 different compounds, such as isoamyl acetate, already described by Butler in its effect, besides butyl acetate, 1-hexanol, 1-butanol, 1-octanol, hexyl acetate, octyl acetate and 2-nonanol. These components have low molar mass, are volatile, and are the most nonspecific of all pheromones. Alarm pheromones are released when a bee stings another animal to attract and entice other bees to attack. Smoke suppresses the effect of alarm pheromones, which is exploited by beekeepers. 69. The other alarm pheromone of the honeybee contains mainly 2-heptanone, another volatile substance released by the mandibular glands. 70. This component has a repellent effect on predatory insects. The alarm pheromone cocktail of the bedbug contains unsaturated witch and octanaldehydes, which are perceived as a characteristic sweetish odor in bedbug-infested rooms. 70. Rooms. 71, 72. Marking and dispersion pheromones. Certain insects, such as the cherry fruit fly, mark their oviposition sites in such a way that other females of the same species avoid the site and lay their eggs elsewhere to avoid competition for food among the offspring. 73. Territorial social insects, such as colonies of ants, also mark territories they claim with pheromones. 74. The marking pheromones include the dispersion pheromones, with which, for example, bark beetles prevent overcolonization of a tree. 75. The females and nymphs of the German cockroach transmit dispersion pheromones in direct contact via their saliva. In the nymphal stage, these serve to deter adult cockroaches and thus protect against cannibalism. In adults, they prevent the translocation of a habitat. 76. Trace pheromones. Fire ants use track pheromones to mark the path from the colony to feeding sites. Trace pheromones are mainly known in insects living in colonies, which mark their paths with low volatile substances such as higher molecular weight hydrocarbons. Ants in particular often mark the path from a food source to the nest in this way. 77. As long as the food source exists, the trail is renewed. When the food source dries up, the ants spray over the trail pheromone with a repellent pheromone. 78. In 1921, the U.S. naturalist Charles William Beebe reported on the ant mill phenomenon, which trace pheromones can trigger in army ants. If the animals are separated from the main trail of the colony, the blind ants follow the pheromone trails of ants running in front of them. These run in large circles until complete exhaustion or death, without finding their way back to the colony. When the food source dries up, the ants spray over the trail pheromone with a repellent pheromone. 78. In 1921, the U.S. naturalist Charles William Beebe reported on the ant mill phenomenon, which trace pheromones can trigger in army ants. If the animals are separated from the main trail of the colony, the blind ants follow the pheromone trails of ants running in front of them. These run in large circles until complete exhaustion or death, without finding their way back to the colony. 79. Recruitment pheromones. Recruitment pheromones are widely used as an element of chemical communication in social insects and have been demonstrated for bees, termites, and ants. These pheromones are used by insects to stimulate other members of the colony to forage at a food source. 63. Bumblebees perform a dance similar to the waggle dance primarily to distribute recruitment pheromones. 80. Primer pheromones. The order Hymenoptera contains the largest group of eusocial insects, including many bees, especially of the subfamily Apiinae, ants, and some species of Vespidae, especially of the subfamily of Vespides. Characteristics often include the presence of a reproductive queen and castes with specialized workers bees and soldiers. Termites form the second major group of eusocial insects. Colonies are divided into distinct castes, with a queen and king as reproductive individuals, workers, and soldiers defending the colony. 81. Primer pheromones have a major influence on the organization of hymenopteran states formed by Hymenoptera and of termite colonies. These pheromones influence the hormonal system of the recipient. They often interfere with metabolism via a signaling cascade or activate proteins that can bind to DNA. In contrast to the releaser pheromones, the primer pheromones are less well studied. For a long time, only one primer pheromone, 9-ODA, was known. Primer pheromones of bees. A well-known example of primer pheromones are the queen bee pheromones. 82. These pheromones control social behavior, comb maintenance, swarming, and ovary formation of worker bees. The components are carboxylic acids and aromatic compounds. E-9-oxodec-2-enoic acid, 9-ODA, for example, suppresses further breeding of queens and inhibits development of ovaries of worker bees. It is also a potent sex pheromone for drones on nuptial flight. 83. Brood recognition pheromones are emitted by larvae and pupae and discourage worker bees from leaving the hive while there are still offspring to be cared for. Furthermore, they suppress the formation of ovaries in worker bees. The pheromones consist of a mixture of 10 fatty acid esters. Brood recognition pheromones are emitted by larvae and pupae and discourage worker bees from leaving the hive while there are still offspring to be cared for. Furthermore, they suppress the formation of ovaries in worker bees. The pheromones consist of a mixture of 10 fatty acid esters, including glycerol 1, 2-dioleate-3 palmitate. 84. Worker pupae contain 2 to 5, drone pupae about 10 and queen pupae 30 micrograms of the pheromone. Older, foraging worker bees release oleic acid ethyl ester, which inhibits the development of nurse bees and makes them care for brood longer. 85. The oleic acid ester acts as a primer pheromone and stabilizes the ratio of brood-carrying and food-producing bees. The foragers produce it from nectar mixed with traces of ethanol, which they feed to the nurse bees. This delays their development until the number of older foragers decreases and with it the exposure of nurse bees to ethyl oleate. Caste determinant pheromones. The Reticuliderms flabipes uses terpenes such as gamma-catenin and gamma-cateninol as caste-stimulating or inhibiting primer pheromones. These assist the juvenile hormone in determining the position of totipotent workers in the caste system. 86. In ants, female larvae possess bipotentiality for some time and thus the possibility of developing as either queens or workers. At some point in larval development, continued feeding determines the fate of the larva. If the juvenile hormone titer is raised above a certain threshold, gynomorphs develop, otherwise, workers develop. Control of larval feeding is governed by a primer pheromone of the queen ant. 87. Application. In the 19th century, Lymantria dispar escaped from entomologist Eddie N. Leopold Trouvelot in Massachusetts and spread throughout the United States by the mid-20th century, becoming one of the most feared pests today. As early as 1898, Edward Forbush and Charles Fernald made attempts to control the population of the gypsy moth by luring males into traps set with attractant females. 88. The United States Department of Agriculture continued these attempts in the 1930s, using extracts of female abdominal spikes to attract male moths. 89. The United States Department of Agriculture was the first to use this method. The application of insect pheromones in pest control has been intensively studied, especially since the first syntheses, with the aim of developing environmentally friendly methods to control population dynamics. 90. Insect trap for bark beetles. In pest control, the use of pheromones in attractant traps to control insects is common practice. This can involve attracting the insects to kill them with an insecticide or physically to trap them or for monitoring. Bark beetles are attracted with aggregation pheromones to trap them. The attractant is usually released when drilling into spruce wood, signaling that the tree can be colonized. The bark beetle trap is an important tool for controlling bark beetles. 91. However, the use of attractant traps poses the problem that the pheromone may act as a chiromone and thus attract predatory insects. By reducing the population of natural predators of the bark beetle, the pheromone trap has a counterproductive effect in this case. 92. Monitoring by means of attractant traps, such as window traps, is used for the quantitative detection of pests to control them more specifically with insecticides depending on the activity detected. In addition, they are used in the identification of new species. 93. Trap beetles work on the same principle as attractant traps. The bark beetles of the initial infestation attract further conspecifics by aggregation pheromones. Windthrow is suitable as trap beetles, which can be equipped with pheromone dispensers to enhance the attraction effect. Trees prepared in this way divert approaching bark beetles from the stand and bind them to controllable stems. The use of trap trees requires regular inspection of the trees. When larval galleries appear, the trees are debarked and larvae and pupae dry out. If necessary, the infested tree may be treated with insecticides or burned to prevent escape of the next generation. 94. Egg-laying preventing marking pheromones are widely used in the insect world. Various experiments have demonstrated the possibility of controlling population dynamics by these pheromones. 95. Application of the oviposition preventing marking pheromone of Rhagoletis sericea, which cannot be controlled with yellow boards, for example, reduced infestations of cherries by 90%. 96. Another application is the confusion method or mating disruption. Here, a high substance concentration of artificially produced pheromones is applied. This makes it impossible for the males to follow the pheromones of the females, thus hindering the reproduction of the pest. The confusion method has a species-specific effect. 97. It is usually successful with respect to one species if sufficient dispensers are applied, but in some cases related species occupy the ecological niche that is released. Bees use the Nasanov pheromone to guide worker bees back to the hive. The pheromone contains terpenes such as geraniol and citral. Beekeepers use a man-made product to attract bees to an unused hive. 98. The process is suitable for trapping Africanized honeybees in trap boxes. 99. Toxicology. Toxicological studies were mainly carried out in connection with the approval of pheromone traps and dispensers. A health hazard cannot be generally assessed due to the large chemical diversity of pheromones, but is usually excluded because only small amounts are emitted. However, in higher doses, orally administered pheromones such as cantharidin cause death in rare cases. 100. Evidence. Recording of an electroantennogram is a function of scent current concentration. Commercial application in crop protection intensified the study of pheromones and led to the development of highly sensitive analytical methods. 32. The identification of a pheromone proceeds through several steps. First, an extract of the pheromone is obtained. This is done by the method already used by Boudinant of extracting glands or whole animals with an easily evaporated solvent, ideally at the time of high pheromone production. Alternatively, the pheromone is adsorbed on activated carbon from the gas phase and an extract is obtained with little solvent. 101. For very small traces, solid phase microextraction is suitable. For identification, the extracts or the solid phase microextraction samples are analyzed by gas chromatography mass spectrometry. 101. The electroantennogram technique is suitable for studying the biological activity of insect pheromones. 55. 102. In this technique, an electrode inserted into the antenna main stem and an antenna branch measures the change in electrical voltage as a function of the concentration of pheromone molecules impinging on the antenna, which are transported to the antenna in a defined manner by an air current. 58. By varying the pheromone molecule, the influence of certain functional groups interacting with the chiral elements of the receptors can be determined. 55. The coupling of gas chromatography and electroantennogram allows the biological activity of the compounds present in an extract to be verified. Insect ecology. Insect ecology is the interaction of insects, individually or as a community, with the surrounding environment or ecosystem. 1. This interaction is mostly mediated by the secretion and detection of chemicals, semiochemical, in the environment by insects. 2. Semiochemicals are secreted by the organisms, including insects, in the environment and they are detected by other organisms such as insects. Semiochemicals used by organisms, including insects, to interact with other organisms either of the same species or different species can generally grouped into 4. These are pheromone, cinnamone, allomone and chiromone. 3. Pheromones are semiochemicals that facilitates interaction between organisms of same species. Cinnamones benefit both the producer and receiver. Allomone is advantageous to only the producer whilst chiromones is beneficial to the receiver. 3. 4. Insect interact with other species within their community and these interaction include mutualism, commensalism, amensalism, parasitism and neutralisms. 5, 6, 7. A giant water bug attacking a fish. Insects play significant roles in the ecology of the world due to their vast diversity of form, function, and lifestyle. They are the major contributor to biodiversity in most habitats, except in the sea. They play a variety of important ecological roles in the many functions of an ecosystem. In the case of nutrient recycling, insects contribute to this vital function by degrading or consuming leaf litter, wood, carrion and dung, and by dispersal of fungi. Insects form an important part of the food chain, especially for entomophagous vertebrates such as many mammals, birds, amphibians, and reptiles. Insects play a critical role in maintaining community structure and composition, in the case of animals through diseases transmission, predation and parasitism, and in plants through phytophagy and plant propagation through pollination and seed dispersal. 8. From an anthropocentric point of view, insects compete with humans. They consume as much as 10% of the food produced by man and infect 1 in 6 humans with a pathogen. 9. Community ecology. Community ecology is the process by which a group of organisms which live in the same location interact. There are indirect interactions, such as reproduction, foraging patterns, and decaying. There are also direct interactions, which take the form of symbiosis, competition, and predation, which are the most easily notable. 10. Every organism at its most basic state could be a consumer in some situations, and a producer in others. The culmination of all these interactions is what defines a community and what differentiates one from another. Insects often play numerous roles in these communities, although these roles vary widely based on what species is present. Insects recognize their host source of food by means of their visual, olfactory, gustatory, and tactile cues. 11. Decomposers. Dung beetles, Scarabaeus laticollis, and dung ball. Decomposer insects are those that feed on dead or rotten bodies of plants or animals. These insects are called saprophages 12 and fall into 3 main categories: those that feed on dead or dying plant matter, those that feed on dead animals' carrion, and those that feed on excrement feces of other animals. As dead plants are eaten away, more surface area is exposed, allowing the plants to decay faster due to an increase in microorganisms that eat the plant. 13. These insects are largely responsible for helping to create a layer of humus on the soil that provides an ideal environment for various fungi, and microorganisms. These organisms produce much of the nitrogen, carbon, and minerals that plants require for their growth. Carrion feeders include several beetles, ants, mites, wasps, fly larvae, maggots, and others. These insects occupy the dead body for a short time but rapidly consume and/or bury the carcass. Typically, some species of fly are the first to feed on the dead body, but the order of insects that follow is predictable and is known as the faunal succession. Many dung beetles and dung flies are attracted to the smell of animal feces. The adults often lay egg on fresh excrement and the larvae will feed on the organic matter. Many species of dung feeders have evolved and only feed on feces from a specific species. There is even a species of dung beetle that will roll feces into a ball, push it into a pre-dug hole, lays egg in the dung, and then covers it with fresh dirt to provide a perfect nursery for its larvae. Carnivores. Carnivorous insects survive by eating other living animals, be it through hunting, blood-sucking, or as an internal parasite. These insects fall into 3 basic categories: predators, parasites, and parasitoids. An elongate twig ant. Pseudomyrmex gracilis, attacking a young mantis. Predatory insects are typically larger as their survival is dependent upon their ability to hunt, kill or immobilize, and eat their prey. 14. However, there are several exceptions, with ants being the most notable. Ants, and other colony insects, can use their sheer numbers to overwhelm their prey even if the ants are significantly smaller. They often have specialized mandibles, mouthparts, for this task, some causing excruciating pain, paralysis, or simply having a high bite force. Conversely, insects that live on their own must be able to reliably bring down their prey and as such have developed a myriad of unique hunting methods. Some actively travel, in search of prey, while others wait in ambush. Others may release chemicals to attract certain creatures, and others will eat anything they can. 15. A fly carrying a parasite. Parasitic insects live on or within their hosts. The parasite causes the host some harm, but not enough to kill it. The presence of the parasite is often not noticed by the host, as the size discrepancy is typically so vast. Parasites vary widely in how they survive in or around their hosts. Some complete their full life cycle within the body, such as the females of most Strepsiptera species, while others may only stay in for the duration of their larval stage. Kleptoparasites obtain food by stealing it from their hosts. A kleptoparasite may opportunistically feed on prey that has been recently killed by a predator, such as many adult freeloader flies, or it may deceptively live in the host's nest, such as the majority of the ant crickets. There is as great a variation in methodology and species in parasites as in any other type of insect. The most threatening parasites to humans are ones that live outside the host and consume the host's blood. These species transmit viruses, disease, and even other, smaller parasites to the host, spreading these throughout the populations of many third world countries with poor health care. Female golden hunting wasp dragging paralyzed spider into its nest. A subcategory of parasites known as parasitoids. A parasitoid is an organism which develops on or in another organism, host derives its nourishment from the host and eventually kills the host. 16. In insects, a parasitoid is an insect whose larva grows by feeding in or on another arthropod, host, and eventually killing the host. 17. 18. The majority of parasitoids insects consume their victims as larvae, while the adults often feed on nectar or other organic material. One family of wasps, the spider wasps, will paralyze spiders before bringing them back to their nest and laying an egg on the spider's abdomen. Other parasitoid wasps, such as ichneumon and braconid wasps, lay their eggs on or directly inside of their hosts. Many of the adult female wasps have long ovipositors, which may be longer than the entire body length of the adult. Parasitoid beetles in the family Rhipiphoridae attack various types of insects, as do most members of the large family of tachnid flies. Herbivores. A phytophagus, herbivorous insect. Out of all described eukaryotes, almost 1/3 are herbivorous insects, about 500,000. 19. They feed on living plant matter or the products of a plant. They are also called phytophagous insects. These insects may eat essential parts of the plant, such as the leaves or sap, or they may survive on the pollen and nectar produced by the plant. These insects will compete with other organisms for limited plant host in an environment where there is constant change in plant availability and quality. 20. Herbivorous insects often use olfactory or visual cues to determine a potential host plant. A visual cue could simply be the outline of a certain type of leaf, or the high contrast between the petals of a flower and the leaves surrounding it. These are typically associated with the olfactory signal an insect may receive from their intended meal. The olfactory cue could be the scent of the nectar produced by a flower, a certain chemical excreted to repel unwanted predators, or the exposed sap of a cherry tree. Either of these two senses could be the driving force behind an insect choosing to consume a certain plant, but it is only after it takes the first bite, and the confirmation of this food is made by its sense of taste, that it truly feeds. Since most insects depend on plant as their source of food, plants have developed some defensive mechanisms to protect themselves from insects. These mechanisms are largely grouped in two: antibiosis and antixinosis resistance. With antibiosis, the defensive trait of the plants affects the growth, survival and development of the insect, but with the antixinosis defense, chemical and morphological factors affect the feeding behavior of plant. 22. Plants have evolved to produce several secondary metabolic substances to protect themselves from herbivores' insects. These chemicals are grouped into alkaloids, terpenoids and phenolics. Insects also have developed mechanism to detoxify these chemicals produced by their host plants. 11. After a herbivorous insect is finished feeding on a plant, it will either wait there until hungry again, or move on to another task, be it finding more food, a mate, or shelter. Herbivorous insects bring significantly more danger to a plant than that of consumption. They are among the most prominent disease-carrying creatures in the insect world. There are numerous diseases, fungi, and parasites that can be carried by nearly any herbivorous insect, many of which fatal to the plant infected. Some diseases even produce a sweet-smelling, sticky secretion from the infected plant to attract more insects and spread farther. In return, plants have their own defenses. Some of these defenses are toxic secondary metabolites to deter insects. These toxins limit the diet breadth of herbivores, and evolving mechanisms to nonetheless continue herbivora is an important part of maintaining diet breadth in insects, and so in their evolutionary history as a whole. Both pleiotropy and epistasis have complex effects in this regard, with the simulations of Griswold 2006 showing that more genes provide the benefit of more targets for adaptive mutations, while Fisher 1930 showed that a mutation can improve one trait while epistasis causes it to also trigger negative effects slowing down adaptation. 19. Schoenhoven and Associates, from Blaney et al. 1985 to Schoenhoven et al. 1992, illuminate the interplay between chemoreceptor stimuli in Lepidoptera and Orthoptera. They used Helicoverpa armigera, Spodoptera littoralis, S. frugiperda, Chloridia virescens, and grasshoppers. They find that most insects respond immediately and roughly equally to phagostimulant, indicating good food, and phagodeterrent, indicating a food to be avoided, or a material which is not food, substances. They also present some divergent examples, both delayed response, suggesting that food decisions were mediated by cognition and not just simple chemoreception, and unequal chemoreceptor stimulation, with gustatory cells firing equally when presented with any material, but deterrent cells firing to a greater degree for undesirable materials. They also investigate similar questions of seeking avoidance in common questions of dietary balance of protein and carbohydrate, i.e. less risky dietary choices where toxins are not the deciding factor, and find similar results, with some insects eating solely by chemoreception and some showing delayed decisions, suggesting cognition. Both salicin and caffeine are antifeedants, and some of the Schoenhoven group's investigations test both the deterrence they produce and habituation to them. The Glendening group has done some similar work. They find Manduca sextus habituation to salicin to be cognitively mediated because deterrent sensory cell stimulation barely decreases even when avoidance ceases. On the other hand, Glendening et al. 1999 finds M. sexta habituation to caffeine to be due to change in chemoreceptor activation because it decreases significantly, and at the same time as cessation of feeding avoidance. The same work tests the cross effects of habituation between the two chemicals, finding that they probably share a second messenger. For both phagostimulus and deterrence stimuli, they find that the effects of multiple stimulations by multiple substances, upon the same cells, simultaneously, produce additive effects, up to the cell's firing rate ceiling. 23. Climate change is expected to change herbivora relationships. Liu et al. 2011 finds no change in distribution in one example, but instead the same herbivore switched primary hosts due to altered flowering time. Gillespie et al. 2012 found host mismatch due to temperature shift. These methodologies in herbivora could be applied to study the same question in climate change plus pollination. As of 2014, however, this remains to be tried. 24. Interspecific relationships. Due to their diverse functions, diets, and lifestyles, insects are integral components of terrestrial ecological communities. Beyond functioning as decomposers, carnivores, and herbivores, insects often participate in other species interactions such as symbiosis. These interactions can both positively and adversely affect plants, mammals, and other insects. These relationships are mostly symbiotic relations. 25. Symbiosis. Symbiosis 26 is a broad term that encompasses various types of long-term interactions between different species. Symbiotic relationship exists between insects and other organisms. It has been demonstrated that the symbiotic relationship between insect and microorganisms is a common phenomena. 27. While mutualism and parasitism are specific types of symbiotic relationships, there are other forms as well. For instance, in some cases, insects may engage in symbiotic relationships where one species benefits while the other is unaffected. This is known as commensal symbiosis. In other cases, obligate symbiosis is a specific class of insect symbiosis that plays a crucial role in overcoming nutrient limitations. In this type of symbiotic relationship, many insects rely on microbial partners for nutrient supplementation. 28. Pollination of a flowering plant by a bee. Mutualism. Mutualism is a symbiotic relationship between two species in which each benefits. 27. In this type of relationships, each species involved provides some services to the other species, and this could be nutrition, defense, etc. 29. Signaling is known to play a major role in selecting mutualistic partners. Odors and chemical detection is involved in the formation of symbiotic relation between most fungus growing